Have you ever seen ants walking in a line and wondered how they do it? Scientists have known for a long time that ants use secreted chemicals called pheromones to communicate complicated social signals, including which trails to walk on. While we knew that ants fluently speak pheromone, we didn't know what specific genes and receptors they use to perceive those pheromones. A new paper from researchers at Rockefeller University has uncovered the genetic basis of pheromone detection, and they did that by creating genetically modified ants. They learned that pheromone detection is all about smell, and a class of proteins called odorant receptors. These researchers had two goals in mind. The first was to figure out what receptors the ants were using to perceive pheromones. The second was to see just how weird these ants would be without them. Ants have about 350 genes in the odorant receptor family, which is many more than other types of insects like flies or bees. Because they have so many more receptors, and because pheromones play a huge role in the life of an ant, these researchers thought that odorant receptors may be involved in pheromone perception. But how can you test whether odorant receptors are required to perceive pheromones? If you had to individually shut down all 350 receptors to find out, it would take a lifetime. Luckily, there is one protein that all odorant receptors and only odorant receptors need to function. That protein is called orco, and it's the protein that these researchers mutated in order to make their ants. Ants without orco lack all odorant receptor function, allowing these researchers to use orco mutants to study the role of odorant receptors in pheromone perception. While making an ant lacking orco may sound easy, it's definitely easier said than done. For many ant species, creating a mutant is practically impossible to do. These researchers got around that problem by using a special ant species called the clonal raider ant. They're called clonal because they make exact duplicates, or clones, of themselves to keep up the population. This clonal colony structure means that getting a mutant ant is much faster than with ants that have a sexual reproductive system. Alright, so now we know which ant they used and what gene they mutated. So let's get to the question I'm sure you're all wondering. How weird